Welcome back to Monkeys on Typewriters on YouTube. We are talking about this big question: how does this end? Thoughts on the evolutionary trajectory of the ongoing SARS-CoV-2 pandemic. And in the format similar to what we've done with the cancer series that we also recommend you check out, Arijit is going to give a presentation. This is one first one in a series that we'll be doing on this topic, and then it'll be followed by a little bit of back and forth discussion. So we'll have links to the other parts of the series and the cancer series in the description below, and hopefully you'll check those out too. Arijit?
Thanks, Ryan. So we find ourselves at an interesting moment in time, right? We basically stopped talking about COVID. There's talk about bird flu, but in general, a lot of people have been writing articles in the newspapers about what did we learn from COVID 5 years ago, and so it's very much been not only relegated to the past, but a lot of what we've heard has been, well, the lessons learned were this, and this. And often those lessons are, well, we did too much to prevent infectious disease. And often what we've sort of seen both in both the experts and the government say is, we need to learn to live with infectious disease. And this is a sentiment that comes up sometimes in so many words and other times in the actions. And so then the question becomes, well, where does this whole "we have always lived with infectious disease" come from, right? And have we actually always lived with infectious disease? So this is the Children's Cemetery in Baltimore, and there are cemeteries like this everywhere, children's gravestones. And these are all like 19th century Victorian things. And you go up to them, and there's like sometimes there's rows and rows of dead children. And this was very standard. It's not uncommon in any graveyard in North America. And so the point is, you know, why is it so common? And the answer is because about 10 to 20% of children born in that era did not live till the age of 5. So when we say we've always lived with infectious disease, we're really saying we've always died with infectious disease. And that was the signal achievement of modern medicine, 20th century medicine, the idea that you didn't have to have 5 kids in order to see one or two grow to adulthood. So if we want to learn to live with infectious disease, this is what we would have to accept because this is what it looks like. And that I think is an important point to frame this conversation is, yes, we have lived with infectious disease in the past, but it mostly involved a lot of dying. Um, and so, we're going to talk today about COVID specifically. Um, and of course, those who forget history are condemned to repeat it. This is actually a picture I took on the Washington Mall in 2021. And at the time, they had this whole field of little white flags, and this was the quote unquote COVID memorial. It's gone now, of course, but so it was literally a temporary memorial. Um, and so today we're going to be talking about that sort of overlap between pandemics and emergent diseases and how that factors into sort of what our thinking should be on COVID. So we'll break this down and really sort of ask the question: what lessons do both history and biology have for us at this point? So we can break that down into a couple of different pieces, and this is a series. So in general, we'll be going through this, one at a time. But today's will be the first of 4 or 5 talks, and today's one will focus narrowly on the question of biology. So we'll talk about what biology teaches us about the emergence of novel pathogens in this presentation. Subsequent presentations will cover the topic of what does history teach us about it, which is a different lens. Then we'll ask the question, what happens next with SARS-CoV-2? What does the research show us in terms of how predictable was it? How predictable do we think it will be in the future? And you often hear people saying, oh, you can't predict all these things or the science change. We'll see that actually a lot of this was predictable, and the predictions that can be made today are not very exciting, to put it mildly. And then we'll also talk about how to win against SARS-CoV-2, because obviously we're in this and it's not gone away. And if we're looking for a future that doesn't involve burying 10 to 20% of children, I'm not saying specifically for SARS-CoV-2, but once you move to to a future where you're accepting all forms of infectious disease, you're going to see all forms of infectious disease. That's how it goes. In the same way as if you accept weeds in your backyard, there will be weeds in your backyard. So I think that is just a fact of life, and we can talk about if there's a different way to go about this. So as we talk about what biology teaches us about the emergence of novel pathogens, I mean, there's lots of articles out there on topics like, newspaper articles as well as scientific papers about this topic and sort of what does biology teach us about what we can expect with COVID. And I think, the point is that emergence events where a new pathogen shows up into a brand new population, these are rare in the historical record. So there's two different ways to draw inferences, and one is to do deduction to get a sort of an idea and then chew on that idea and come up with an inference drawn from it. So if you say all men are mortal and then you observe that Jason is a man, then you will realize that Jason will die at some point, right? On the other hand, induction is the other way to do this. You take a whole bunch of observations and then you draw a general rule from them, right? So for example, you could say, look, every time I eat peanuts, I get this nasty rash. Eventually you realize that being a symptom of allergies implies that you are allergic to peanuts. So induction and deduction are a little bit different and they are used differently. So when you're talking about deductive reasoning in the specific context of what you can draw as inferences from COVID, you may say this can or this might happen. Whereas for inductive reasoning, if you look at a lot of different pandemics, you could say this always happens or this never happens. So that these are not necessarily what inductive and deductive reasoning are in every case. But in the case of COVID, there's two ways to reason. One is to say, I see a thing, and from that I can deduce that a certain thing will happen. Or, oh, I've looked at all these other pathogens, and this is what we've never seen, or this is what we've always seen. So therefore it will always happen. Because there are so few historical events, this second line of reasoning is deeply fraught because just because you haven't seen something with 5 pandemics doesn't mean you won't see it on the sixth. Whereas deductive reasoning, if you've seen something in a previous pandemic or you've seen something in a previous emergence event, then yeah, there's a good chance you may see it again. So this is a key tenet of risk management. You look at things that could go wrong and you plan for them. And you can plan for things that have never happened. But if things have happened already, you certainly should be planning for them. You shouldn't be ignoring them. So at a minimum, the low bar for risk management is look at the things that have happened before and say, okay, if that happened again, how screwed would we be? Um, to use inductive reasoning and say, okay, this thing has never happened before, therefore it won't happen now. That's just calm mongering. So we wrote an article, you and I wrote an article on calm mongering. And I think this is something that, by the way, folks will share the link with you below this. And it's an article worth reading because there is a whole bunch of pseudoscientific techniques that people use to make you believe as if all is well. And sometimes it is, and sometimes it isn't. And it's important to understand when science is being manipulated in that way, often by scientists or by medical professionals. So part of this just comes down to how people draw inferences. And when you are drawing inferences with motivated reasoning, you can draw some really, pardon my French, but shitty inferences. One such inference was drawn by the volcanologists. There's a whole field called volcanology, who were performing risk assessment on the risk of eruption of Mount St. Helens in 1980. Now, at that time, what they would do is they would say, okay, well, no two volcanoes are exactly alike. So in order to understand what's going to happen at this volcano next, we have to look at its previous eruption history. So if no two volcanoes are exactly alike, or you cannot draw any inferences from one volcano that you can generalize to the next one, then you should get rid of all volcanologists because they're actually not useful. You could simply have a historian sitting there taking notes and telling you what's happened in the past. So the whole point of having a field about a topic like volcanoes is you should be able to draw generalizable inferences. Otherwise, don't bother. So of course, when they went down this route, it was obviously like intellectually flawed, but it's not an abstract thing that it was intellectually flawed. What ended up happening was there was a giant pool of magma, like a half-mile wide, forming under the volcano. And it was so wide that it was actually cracking the surface of the volcano, and the volcano was distending. Now, common sense would tell you that something might happen as a result of a mile wide or half-mile wide pool of magma sitting right under the surface, struggling to get out. But these guys were like, well, we've never had an eruption of that type at this volcano before. So essentially, they were like, well, don't worry about it. Uh, of course, the rest is history. The whole point being that just because something hasn't happened before doesn't mean it won't happen. And ignoring common sense will not lead to a good outcome. So, at the end of the day, what St. Helens really was able to point out is that, you need to do risk management on the basis of things that could happen. And this is a key point I think that a lot of people miss, is that when you're doing risk management, you really have to be able to assess it in the context of possible outcomes, and not sort of dismiss them because you haven't seen them happen in this very specific context. You'll never be able to generalize if you always insist on the specific context. So this point about risk management has been made by folks such as that book, The Failure of Risk Management, which is a good read, and also by Nassim Nicholas Taleb in his book, The Black Swan. And in his book, The Black Swan, Taleb talks about this interesting thing, which is essentially this concept of unprecedented risk. So essentially, this thinker Al-Ghazali pointed out that again, that something hasn't happened. Just because something hasn't happened doesn't mean it won't happen, right? So, if you think of it, a turkey, right? When a turkey lives on a farm, it can live its whole life with the farmer being a benign presence in its life. And so the turkey is going to think, farmer's a good guy, farmer's on my side, farmer cares about me. The week before Thanksgiving, the turkey is going to find out that this is actually not the right way to think about the situation. And what you end up with is called a revision of belief, right? And that revision of belief can have violent consequences. And so the point is that a black swan event is only a black swan event based on your failure to anticipate the future very often. So, in this particular case, the turkey is not able to look forward and say, well, why is the farmer feeding me, or what is the outcome that is likely? Anthropomorphizing the turkey. I don't know what turkeys think about in their heads. I haven't really spent a lot of time thinking about it, but the point is, if you refuse to acknowledge the entirety of the context when managing risk, and you focus on the things that can't happen because they've never happened before, you could meet a very sticky end. So, with that sort of aside out of the way, let's talk about what biology teaches us then about the emergence of novel pathogens. Um, and so we know this kind of intuitively, that when pathogens emerge from animal species into humans, they go through certain steps. So, for example, rabies is only in animals, it can infect humans, but it doesn't go any further. Something like Ebola basically infects humans, but then those infection chains can propagate to some distance. Something is like there are diseases like dengue, which basically keep reinfecting or have a pool back and forth between animals and humans. And then there's diseases that like HIV, which jump from animals to humans and now are propagating only in humans and freely. So when you have this kind of a stepwise sequence, the question becomes sort of, where in that stepwise sequence are the changes happening, and what impact do those changes have on virulence? What impact do those changes have on outcomes? So it begs the question, in this change, in this step of sequences, if you let a disease keep running for long periods of time, what changes can you expect? And so it turns out that when a pathogen jumps from one species to another, virulence can either increase, can remain unchanged, or decrease. So, for example, if you have a virulence, an increase of virulence, you have a disease which is relatively benign, for example, in chimpanzees, jumps to humans, becomes deadlier. Um, Similarly, you could have the opposite happening, which is that the disease on the right makes a species jump and then is actually less virulent in the new species. So biology has examples of all of these different things. That's not how people usually think of it. They think about it in terms of the law of declining virulence. It gets said a lot, it has gotten said a lot during this pandemic that, over time, COVID will become more manageable because of evolution, quote unquote. And the idea is that where does this idea come from? It comes from uh, a statement made about 120 years ago by this guy, Theobald Smith, who was actually a museum curator. And his point was, his self-taught natural historian, his point was, he called it the law of declining virulence. The term law has a very specific meaning when it comes to biology, but this was just his opinion. And so his point was pathogens tend to evolve towards becoming less virulent over time when they coexist. Because if it's too pathogenic, it will kill or incapacitate its host. This is a neat idea. Um, it's compelling. It's kind of exciting to think about. Well, just let diseases run and they'll eventually get tamed. Um, Unfortunately, it's not supported by data. And again, the guy wasn't a scientist. He was a natural historian and a museum curator. So fair enough, good for you that you came up with an idea. Scientists in the century and change that has since gone by have examined that and looked to see what happens. So here, for example, is an experiment that people did to ask this exact question. And what they did was they took a virus, tobacco envelope virus, TB, which infects Nicotiana tabacum. And they then had it passaged in two different species that it can also infect in the species jump. One is Nicotiana benthamiana, and the other is Zetorastramonium. And one is closely related and one is not so closely related. And they asked the question, now in these plants, what would happen? So they did the competition assay with the ancestral virus. And they asked the question, when you compete with the ancestral virus, how well does the new virus do how well does the virus do in the new species uh, after multiple generations? So after 4 generations or 5 generations, they would then introduce the ancestral virus back in and see how the new virus was doing after several generations. And what they found on the left is that um, Nicotiana benthamiana, where the virus is actually has much higher virulence, over time, what they found was with multiple generations, um, the virulence actually increases, which is these bars on the right. If you look at the TEV and compared to the ancestral, the two are about the same. There's no change in virulence. But then, as it goes by, you see increases. On the other hand, on the right, what you see is in Deuterostromonium, where basically the virus is less virulent in in this in this new host species than in the original. What ends up happening is that you see, generally speaking, a decrease in virulence. So there's no rule about this, and this is a direct experiment that shows that it bounces around. If you look at this, you say, well, it's not really increasing a straight linear trend or decreasing a straight linear trend. It is bouncing around. But the notion that logic would have said, if we are following that law of decreasing virulence, that you would have seen a strong trend towards reduced virulence in the host species where the virus was more virulent. You did not see that. You saw, in fact, the opposite. Um, that is, that the species where in the host species where the virus was less virulent generally saw some decreases. The host species where the virus is more virulent saw statistically significant increases in virulence. So much for that idea, right? People have looked at this more broadly. And so after the law of decreasing virulence, the second framing of this in the 70s was something called the virulence transmission trade-off. And so the idea was, okay, well, virulence actually increases transmissibility up to a point, and then after that point, it starts declining, and the added benefit of additional virulence keeps decreasing. So again, this is obviously much more rational than the law of decreasing virulence. But the question is, how much do you observe this? So what these guys did in this paper in a meta-analysis was they fit a simple line, which is a polynomial, y equals b naught plus b1x plus b2x squared. So the b1x is a straight line, and the b2x squared part is the part that will curve down. And what they did was they asked the question, now let's take a look at different data sets and ask the question, how well can we fit this across experimental evolution studies looking at this question? Um, so what they found was that when they looked for direct quantitative support for the linear increase in transmission versus the tapering off due to increasing virulence, they found that the linear increase in transmission is pretty well supported. So across a range of different studies, they found that B1, the parameter that suggests a linear increase in transmission, is statistically significant in nearly all of them. So yeah, it's true. Virulence does increase transmissibility experimentally. On the other hand, when they looked for the tapering off effect on the right, the B2 effect, which is this part of the curve, they were not able to find it. So, which goes to show you that at least one part of the hypothesis is true, that increasing virulence increases transmissibility. But the piece about that rate of increase of virulence tapering off is not true. The idea that virulence will lead to decreases in virulence will lead to improved transmissibility is not consistent with what is observed. So that's kind of interesting, right? Because in fact, when people talk about virulence and transmissibility, the idea that comes to everyone's mind is this example from myxomatosis, right? So myxomatosis is a famous example. they introduced myxomatosis, which is a virus that infects Latin American lagomorphs, which are rabbit relatives. They introduced it into England and they introduced it into Australia to control the rabbit populations back in the 1950s. And so myxomatosis wiped out these rabbits initially. So you had a huge change in rabbit populations because of this virus that they had never seen. But an interesting thing happened, and this, for example, is from Australia in the 1950s. An interesting thing happened over time. So each of these bars is a mean and standard deviation for the mortality that was observed with myxomatosis in that country um, over time. And what they found was in the first few years or the first few outbreaks of myxo, what they found is that that virulence decreased in a pronounced way um, by about 5 or 8 years after it had been introduced. Now, it's worth noting that rabbits in the wild live for about a year. So you're looking at many generations. And so this is definitely evolution going on. Um, and the idea was, okay, well, this has to be evolution on the viral side, right? The virus evolved to become milder. That was the initial sort of thought process, and it was support for the law of declining virulence. And then people dug into it a little bit in the subsequent years. So this is a 70-year-old source. There's been lots of time to dig into it. And as a more recent paper shows, what ends up happening with the myxomatosis virus is one part of the story. What ends up happening in the rabbits is another. So this paper looked at allele frequency changes in rabbits, wild rabbits that were hosts to myxomatosis. And what they found, they had samples of wild rabbits from essentially, 6 or 7 decades. And what they were able to find was that there were distinct allele frequencies that were changed over time, implying selection in the rabbit population. For example, in interferon alpha 21A, which is an immune system gene, um, that is involved in how strong a reaction rabbits have. Um, that particular allele changed, and so what ended up happening was you had that allele frequency increase over time after the introduction of mixed overdoses in a very clear-cut way, both in Australia and in the UK. Um, and then around the year 2000, when a different virus was introduced into rabbits, that allele frequency fell off. RHDV is a different virus unrelated to myxomatosis. So what you can see, though, in the era that Mixo was being used primarily to control the rabbits, you were seeing allele frequency changes in the rabbits. The rabbits were evolving. During this period, also, the apparent virulence of Mixo decreased across the board in the general case. But what's interesting is that this wasn't the case for all strains. Some strains of Mixo actually became more virulent. So this is the severity grade of virulence. They grade it from 1 to 5, and basically that involves the case fatality rate as well as the survival time. And what they found was that basically when you look at myxoma virus on both continents, now not the rabbits, but the myxoma virus, what they found was that the percentage of field strains with each virulence grade, or percentage of viral strains that were identified from wild rabbits in the field with these different grades was kind of interesting. What they found was over time, between 1962 and 1975, grade 2 strains became more common. Um, and then on the other hand, um, so that made sense. So you were seeing a decrease. But then in the next period of time, you began to see from grade 2 to grade 3, you began to see a shift in the opposite direction. So now you ended up with strains that had essentially greater virulence over time. So that was not expected. Why was the virus becoming more virulent? Um, so people have studied this at length, and they've done big, phylogenetic trees like this of virulence. And the answer is, it's not always the case that it becomes more virulent or less virulent. The red ones here are more virulent, the blue ones are less virulent over time, with the date being provided behind that slash mark. So what you can see is, depending on the year, it's kind of all over the place. When you compare it to the ancestral strains, there are some trees within which virulence has gone up and down, some trees in which it's remained generally the same. It's really kind of random. It's all over the place. And again, the rabbits were evolving to become more resistant. The virus by itself, when used to challenge lab rabbits, was sometimes less virulent and sometimes more virulent. So there was evolution happening on both sides, and the apparent decrease in virulence in myxoma being used to control wild rabbits had a lot to do with the evolution that was happening on the rabbit side. It didn't have anything to do with what was happening on the viral side, where the virus was experimenting with strategies. And sometimes as those strategies led to the virus being more virulent and other times less virulent. But the virus was not doing the rabbit any favors. The virus was trying to figure out what worked. And sometimes what it hit upon was a strategy that involved increased virulence, as you can see over here, all the way into 1991. And in other cases, it involved decreased virulence. Um, so this is an important point. And this happened because virulence was going, the resistance in rabbits was going up. So the virus had more headroom to experiment with strategies that would lead to increased virulence in this population because the population was already more resistant. And so it turns out that this story is actually a longer story than this, but it turns out that one part of this is the different paper looking at the same thing. And again, they showed increases and decreases in virulence over time. But it turns out that one part of the story was that some of the viruses were evolving delayed virulence. So instead of killing rabbits right away, they were just as deadly, but that didn't kick in until 28 or 30 days later. You can see a strain with rapid virulence on the bottom right, where essentially that's a survival curve. By day 20, all of the rabbits are dead. And then on the right-hand side, you can see one with a more delayed virulence phenotype, where it takes about 30 days for the rabbits to die. So this is kind of interesting, right? Because it says that what you're taking away from the myxoma story has two components to it, the viral evolution as well as the rabbit host evolution, and that the virus is actually using that increased host resistance to come up with other strategies, including that of delayed kill. Uh, and so all of these things are very relevant. When we look at the mixer story, all of these things are very relevant. I'm sure you can see the parallels in the COVID situation. And so this was what they pointed out over here, that it was a distinct and novel disease phenotype, typified by collapse and death. Um, Essentially, something that had evolved in the rabbits, evolved in the virus, and a brand new physiology and a brand new pathology for that disease. So we always think of diseases as having fixed pathologies. That's kind of like our mental model. Well, oh, if you have this disease, this and this will happen. But if you look at COVID, the way the virus was in 2020 and the way the virus is today, COVID behaves functionally differently than it did in 2020. So what lessons can we learn from Mixer? So one is that host population resistance makes a difference. And two is host resistance opens up the option of the virus then evolving higher virulence as well. Now remember, in order to get host resistance happening, it happens over many generations. So the rabbits were one generation a year. So it takes many generations, obviously, for that to happen. And when you have host resistance, you have essentially higher, higher virulence as one option. Note that it wasn't a uniform increase in that space. So it's an arms race. It's neither attenuation nor is it a drive towards increased virulence. And basically, disease pathology and virulence are up for negotiation at every step along the way. The virus does something, the host does something, and it goes back and forth in this way. So you can think of it sort of as in a big picture way, you can think of it when a virus emerges, it either drives the host population extinct if it shows up in a new population. Assume that doesn't happen, then it's established in the population. If it's established in the population, then you have some kind of mass mortality event. And that either leads to extinction or elimination of the virus. And you have cycles of evolution. So this sort of repeated cycle of evolution thing going on is the Red Queen's race, which is basically um, it's an ecological and evolutionary concept that deals with the way in which two species evolve with each other. And so the Red Queen's race gets really messy very quickly. And to put it in simple terms, this is antagonistic coevolution. It's an evolutionary arms race, and there's continuous adaptations on both sides. And the host species need to continuously evolve to survive. If it slows down, it can be driven to extinction. Similarly, the virus can also be, or the pathogen more generally, can be driven to extinction as well. So this is a bloody game, and it usually ends badly for one or both of the parties. So that's kind of surprising, right? Because I mean, we always think of evolution as a good thing, right? So anybody who's watched a Marvel movie sort of thinks about it this way. Well, it's easy to see this. Essentially, uh, people talk about evolving in a colloquial way, and the point is that this is not actually how evolution works. People don't evolve. Uh, if evolution is acting upon you, you will simply die or fail to reproduce. That is the only contribution that we as individuals can make to evolution. Um, right. So it's much more Darwin Awards than it is the character Darwin from the Marvel movies. And that's an important thing for people to remember that evolution is not our friend in a very real sense. Um, it's a very vague set of expectations, right? It's like when we think about this step, right, we say, okay, well, we're building up immunity or we're evolving to be resistant, right? And it's kind of like the South Park thing of, the gnomes collecting underpants. And then they say, well, something's going to happen now. And then they're like, well, profit will happen at the other end. The main question is, what is in that question mark, right? You're going to collect a bunch of underpants and then what? So you're going to generate immunity and then what? Like, how are you going to become more evolved? These questions don't really get answered. And we often see this happening in real time when conversations around COVID that, well, the virus is evolving and we'll be fine or whatever. But how will we be fine? What will the virus do? And the important thing to note is that it's absolutely true that acute infections have become a lot less deadly than they were in 2020. So there are virulence changes, but acquired immunity and evolved resistance are two different things. Acquired immunity is when you have an individual that's exposed to a pathogen, and as a result of that exposure to the pathogen, is less vulnerable the next time around. So acquired immunity is an individual level phenomenon. Most of us hopefully have been vaccinated and we have some acquired immunity from the vaccination. Otherwise, you get acquired immunity from infection as well. But that's acquired immunity is an individual level phenomenon. Evolved resistance, on the other hand, works through multiple generations. So what you have is you have exposure to a pathogen in a host population. That pathogen presumably kills off or renders sterile a large proportion of the population, and in doing so, then generates a new population which is now resistant. That's what was happening in the rabbits. It required a lot of the rabbits dying in order for them to evolve resistance. Now, this is silly. It's kind of obvious. Everyone knows this, but we seem to forget it when we're talking about newly emergent pathogens. It's like we will not magically evolve resistance. It's a bloody business, and it takes a lot of dying to get resistance. So evolution is not all fun and games. And sorry, Ryan, I know you study it, but it's not all fun and games. And the point is that the Red Queen's race is not a good game to play. And this is something that people are sort of missing when they talk about newly emergent pathogens, and that is it it's an endless race. So the Red Queen's race comes from Alice in Wonderland, where the Red Queen says it takes all the running you can do to keep in the same place. And so in this particular case, what it means is both the host and the pathogen are evolving. If one of them stops running, it loses the race. So in practical terms, this actually means species level extinction. These things happen. There's many, many well-documented examples of species being driven to extinction or near extinction. This is just a handful, but I could go on and on and on. Um, for those of you who are interested in trees and plants, the the North American flora when it comes to large trees is just a mess now compared to 30 years ago. There's so many species that we used to see all the time that are now hard to find. Ash, hemlock, beech, American elm, obviously. The only American elms you will see around the place are American elms that actually have, um, most of them have, if they've survived, they have tubes in them to prevent Dutch elm disease, which is a fungus showing up. So they literally will put tubes in these trees and feed them with antifungal. Um, so that's kind of where we are now. So we know this as, just kind of intuitively, we know that diseases can drive species to extinction. Um, so then the question is, well, what are the characteristics of diseases that can drive species to extinction? And the answer is those diseases tend to evolve rapidly. They have alternative reservoirs and they tend to be density independent in their spread. So there are many different ways to get density independent spread, but one of them, for example, is airborne spread. There's other ways as well. But the point is, if a disease evolves rapidly, if it has other species it can go live in, if it wipes out one species, and if it can spread, for example, through the air or through the water, then it's got a really good chance of essentially driving one of its hosts or its hosts to extinction. So, for example, one example of this, the classic example, is bulldog rats in Christmas Island. There were these funny-looking rats called bulldog rats in on Christmas Island. And when the first ships showed up, essentially, they brought black rats. They brought European rats in, or Asian rats, as the case may be. And within a short amount of time, within about 8 years, bulldog rats were wiped out. What they found, essentially, they did a bunch of genetics, and they found that very likely this could be pinpointed to a trypanosome. A specific trypanosome was responsible for wiping out these rats. And so what they looked at was they looked to see which of these particular trypanosomes it was, and they were able to go back and literally from samples, from historical samples, find trypanosomes that were found in these rats in a clear-cut way from that era. So it's not cut and dried, but that's really kind of interesting that a specific trypanosome passing from you know non-native rats to native rats was associated with that. Like every single rat sample that they were able to find essentially had that trypanosome in it. Um, and also they disappeared in that 8 year period. So it's kind of interesting when you see that kind of work and that kind of almost forensic pathology that is being conducted on museum samples or being conducted on gravesite samples, for example, is a large part of how we understand how these things are playing out. So it's been very interesting in recent years where sequencing coupled with this kind of forensic approach has really changed our understanding of a lot of these things. So we know that evolution is not our friend, and we know that the Red Queen's race is something that happens when a pathogen meets a host species. So then that begs the question, with those two things together, are humans also subject to evolution? Um, and it's something that is very relevant to us at this point. Like, can we evolve? Uh, so it turns out we obviously can evolve. There were 12 different human species as recently as about a 500,000 years ago or so. We became the sole survivor. And so out of those 12 different human species, the question is, what went on? Why did so many human species go extinct? Um, and what you'll notice is many of these human species had very short lifespans. Many of the actual species had unusually short lifespans. Typically, species will live for tens of millions of years or hundreds of millions of years. Even I think that the average is 2 to 4 million years, 2 to 4 million years for mammalian species. Most humanoid or hominid species have not lived anywhere near that long, and so that's kind of interesting, right? So when we look at human evolution, one of the things we find is that there was a severe genetic bottleneck about 800,000 years ago. Around 800,000 years ago, the total number of humans on the Earth, archaic modern humans, and this is relatively recently, dropped to about 1,200. So everybody is kind of related to each other. Everybody's kind of like a cousin once you're down to that number almost. And they were able to infer this from DNA markers on people, on wide samples of human DNA. And so that's kind of interesting, right? So when they actually went backwards and they said, okay, well, what is the divergence? What is the genetic divergence among chromosomes? They were able to back into it with a phylogenetic model and find that roughly speaking, that number around 800,000 years ago, as estimated by multiple different algorithms, was really, really small. And initially they thought they had the wrong answer. They did this several times and they found this was the answer, that at one point there was a huge bottleneck. So 800,000 years ago is actually very much in Homo sapiens territory. We're looking at Homo erectus, Homo heidelbergensis in that line. And so these are, they're not humans, but they damn well look very similar to us. Um, and so 800,000 years ago is recently enough that it begs the question, could it happen again in some, recent amount, in some reasonable amount of time, so to speak? And what was the reasoning for it? So one question that people asked was, well, could it have been climate change? So it turns out that if you look at climate over the past 800,000 years, you get all these fluctuations. And 800,000 years ago, there was a spike in change in temperatures. But it's not like each one of these other spikes was accompanied by an extinction of humans. So 3 degrees, while bad, or in this case, sorry, 12 degrees, while bad, um, was not bad enough necessarily to be, you can't necessarily attribute this to climate change. There is another candidate, though, for why we saw that bottleneck. And that candidate obviously is infectious disease. So in this case, what people did was they did a they they looked at grave sites out in northern Russia, and they dug up a grave site containing a 12-year-old child from 31,000 years ago. And what they did was they basically sequenced the DNA, and what they found was in the pulp of the tooth, they found evidence for an adenovirus infection. So now using that adenovirus sample plus samples from modern day adenoviruses, they were able to back out evolutionarily what was the rate of change. And what they were able to infer was adenoviruses essentially, in this paper they showed, emerged in the human population about 700,000 years ago. So using that ancient virus, they were able to back into a 700,000 year point of origin for adenoviruses. Now, you'd say, well, that's obviously not going to be killing people, right? I mean, we all know what adenoviruses are. They cause the common cold. Like, what's your point, right? By the way, this was Francois Bayou was one of the authors on this. Those of you who are COVID cautious know his name. Um, it turns out that adenoviruses have left a fingerprint of selection on the human genome. So if you look at the pattern of selection in humans, and if you look at the variation in proteins, you can identify proteins that are under positive selection. The way you do that is you look to see whether the changes in human DNA between people, you look to see whether those changes are preserve protein structure or protein function or not. So if they're present in the third base of each codon, for those of you who remember that, there's 3 bases in the codon that code for a single amino acid. Changes that are present in the third base tend to be synonymous, change because essentially it doesn't impact the protein uh, the protein sequence, whereas non-synonymous changes happen in the other bases. So if you look at that ratio of mutations in the non-synonymous versus synonymous regions of the DNA or of the code for a single protein, you can start trying to figure out whether this particular protein was under selection or not. Proteins that are under selection will have changes that preserve its function. Proteins that are not under selection, you will not see that necessarily. So what they found was that virus interacting proteins in general tend to have a strong degree of non-synonymous, that a very low ratio of non-synonymous to synonymous changes, meaning that synonymous changes are strongly favored relative to non-synonymous changes. And so what they found in this case was for adenoviruses, actually, again, over here, this is the red bar is that pattern for all proteins in the genome. And that's the 95% confidence interval. So you can see adenoviruses lie outside that. And all virus interacting proteins, in fact, when you compare the ratio this is the distribution in red of all proteins, and virus interacting proteins are over here in blue. So most virus interacting proteins have very, very low ratios of non-synonymous to synonymous changes. So why the hell would a common cold be selected for? Like, why would resistance to common cold be selected for? That is actually human evolution. In other words, it's a common cold today because it wasn't a common cold in the past. The benignness of adenoviruses has to do with the fact that, right, you can see right here, it's benign today because it wasn't benign in the past. It's benign today because there was active selection for individuals who had alleles that could resist adenovirus infection. So then you say, well, that's crazy, but I mean, it doesn't kill people, right? So how do you know it ever killed people? And the answer is it's endemic, but it still kills people today. So people with weakened immune systems or existing respiratory or cardiac disease are at higher risk of developing symptoms with a common cold, quote unquote, common cold. Not only that, but you can actually see this in young and healthy people too. So one really good test sample is the US military. In the US military, they mandate adenovirus vaccines. So they're like, everybody talking about why are we giving vaccines for COVID? Because it's just a cold. in the military, they give people vaccines for a cold, and that cold is adenoviral colds. So when they dropped that between the year 2000 and 2011, they saw a massive spike in respiratory infections. So you can see that over here. All kinds of adenovirus infections went up during that period. And once they brought the vaccinations back, it went away. Now you can use the patterns over here to infer what's going on in terms of what impact this has on mortality. And what they saw was in the years when they weren't mandating um, vaccines for this disease, it was regularly killing one or two people every year. And these were young, healthy military recruits who mostly showed up to camp in crowded settings, caught the disease, and died. So every year, soldiers were dying of the common cold um, when they didn't have vaccinations for this. Now you could say this is a negligible amount of people, and that's all fine. But the point being made here is adenoviruses can kill. And obviously, when you have something like this that can kill intrinsically in terms of the biology, these are recruits, so they're all healthy. There's no one in there with sort of a massive number of chronic conditions. Um, and you have the fact that you see a fingerprint of selection on the genome, you can put these pieces together. So remember, we talked about that genetic bottleneck 800,000 years ago. And I pointed out that concomitant with that genetic bottleneck is the era of emergence of adenoviruses. The point being that they emerged at around a period when we saw a bottleneck. As to whether the bottleneck can be ascribed to that, we don't know. But the point is that it's very plausible given that it has a strong selection fingerprint on the genome. So at some point between 700,000 years ago and today, it killed enough people that it left a fingerprint on the genome. And that's why it's a common cold today. And it's still not done selecting for us. If we, you know, everyone says, well, it's a benign thing. Well, maybe it's a benign thing, but how do you know? If we're running the Red Queen's race with an adenovirus today, there's no reason to assume that that Red Queen's race will always play out to our advantage. So what they said is there's no such thing as a benign virus as such. And even for something like adenoviruses, we should probably be coming up with a way to vaccinate for them for the simple reason that they are not benign even today. The military doesn't think they're benign, by the way. And there's no reason to tolerate coexistence with this virus because it clearly killed enough people in the past to leave its impression, to leave a mark on our genome. So moving right along, you know, the idea that endemic viruses become benign out of the goodness of their hearts is not correct. And the idea that they become benign through natural selection in humans is much more plausible. It's a slow and bloody process, and it's probably not a game that we want to keep playing. Um, as another example where humans have been subjected to evolution, we could look at the example of the Neanderthal. So when you look at Neanderthals, the Marjorie Taylor Greene looking lady on the right is a Neanderthal. Um, and so they again look very similar to us, right? And the point is that when you look at Neanderthals and you look at sort of how they sort of spread through Europe and Asia, their trajectory and ours was very similar. They were just separated from us by a few 100,000 years. They spread out of Africa and then they spread into Europe about 750,000 years ago. Modern humans were still in Africa during that period. Then around 100,000 years ago, modern humans radiated out of Africa and Neanderthals disappeared. This is a story. We know that we have pieces of Neanderthal DNA within us. So we know, at least for Asians and for Europeans, we know there was significant interbreeding at some point. So how do these pieces connect? So if you look at the the viral history of the two species, you notice something interesting. There are many viruses, such as adenoviruses, and which we just discussed, and retroviruses, other retroviruses such as polyomaviruses, my bad, um, that humans and Neanderthals both shared. But when Neanderthals evolved outside Africa, there was a whole bunch of new diseases such as Hep B, Hep C, um, and VZV, and smallpox for that matter, that were distinct to humans but not Neanderthals. So when early modern humans showed up in Europe 41,000 years ago, there were diseases that they had that Neanderthals didn't have. Uh, and that's an important point. Now, the two species had diverged from each other for several hundred thousand years. So it was also the case that Neanderthals had diseases that humans didn't have. So you can actually now ask the question, was it a disease thing that drove extinction? So a funny thing happened when humans moved out of Africa. They moved out of Africa about 180,000 years ago. Then for a very long time, about nearly 100,000 years, archaic modern humans were limited. Their northernmost spread was in what is now modern day Israel. And after that, they spread out of that. But the archaic humans spent about 80,000 years in sort of modern day Israel, Syria, Palestine, in today's day and age. And after that, they spread out of there. So what was going on in that zone of contact for 100,000 years? So this particular mathematical model question asked that paper asked that question, and what they hypothesized was that it's possible that that zone of contact was limited by the disease, that humans spreading out of that zone were being felled by Neanderthal diseases, and Neanderthals trying to spread out of that zone in turn were being felled by human diseases. If this is the case, then what you would hypothesize is that there was interbreeding happening. Indeed, there was interbreeding happening in some substantial numbers, it turns out. Um, but that interbreeding had an effect on both genomes in that case. So what you can see over here is that if you look at disease introgression dynamics and you look at the contact rates between the two species over time, what would happen with interbreeding is that over time, both both species were picking up genes from each other. If species one, for example, experienced a lower disease burden and therefore was more enthusiastic about um, shall we say, contact with the other species. Then what you would see is that species one would be picking up disease resistance genes from the other species. So species one is human, species two is Neanderthals here. It turns out, for whatever reason, there was much more human Neanderthal interbreeding happening with Neanderthal females than in the opposite direction. Go figure. But what that means is that humans picked up a ton of Neanderthal genes, while Neanderthals didn't necessarily pick up human genes. This is something we know separately from DNA, from our DNA. So consistent with that, what you would have seen, what the mathematical modeling says, is that species one, which is humans, would have had the Neanderthal disease burden largely dealt with, while Neanderthals had not dealt with the human disease burden, precisely because of that phase of interbreeding, which was somewhat selective. So in that case, what would have happened is that when humans spread out of that zone of contact, they would have been essentially now in Neanderthal territory, spreading disease to Neanderthals while still being resistant to Neanderthal disease. So it's a plausible model. Now, if that model was valid, one of the ways you could test it is you could ask the question, well, are there then a whole bunch of Neanderthal genes in human DNA whose explicit purpose is to interact with viruses or to protect against viruses? And it turns out very much there are. So the introgression of Neanderthal virus interacting proteins with human DNA has been shown. Essentially, the sort of model is that if you have a disease that was deadly to Neanderthals, Neanderthal on the right, stereotypical Neanderthal, what you would expect to see is you'd expect to see that human genes Neanderthal genes that protect against that disease have made their way into human DNA through interbreeding. And that is exactly what you see. So you can actually look for these adaptive introgress segments in human DNA by looking at sort of how often they show up relative to random segments, and you can actually pick them out off the background. Um, and so they did this. This particular paper looked at that, and what they found was if you look at the background, which is in dashed lines over here, and you look at segments of different sizes and segments of different frequencies, what you find is that Neanderthal chunks of DNA from Neanderthals show up in humans at a very non-random frequency. And this is specific to virus interacting proteins. So what they found is when they looked at a list of virus interacting proteins in humans, and they went looking for whether there were chunks of Neanderthal DNA or not, they found yes, indeed there were, both in Asians and Europeans. The gray bands show the 95% confidence interval, and the dashed line shows the background. So you can see that all of these genes at different lengths and at different frequencies in the genome show up where you would not expect to see them on a random basis or on the basis of a bootstrap, so to speak, which is the test for randomness. So yes, indeed, humans picked up DNA from Neanderthals. So that's a hypothesis, and it goes to show you how much of an impact disease has, and particularly viral disease has in shaping evolution. Um, Coming back to that paper I talked about earlier, there are many different viruses for which you can see. This is a general pattern. It's not just adenoviruses. For many different diseases, you can see the examples of virus interacting proteins being under strict selection pressure, purifying selection pressure. So now, not all of these diseases are diseases we consider to be incredibly deadly today. Herpes simplex virus, we wouldn't consider deadly, but hey, by the way, it's left a fingerprint on our genome. Kaposi sarcoma virus rarely kills anyone, bottom line. Again, it's left a mark of selection on our genomes. Influenza has left a mark of selection on our genomes. This goes to show you that these kinds of proteins are there for a reason, and it also goes to show you that basically every one of these diseases at some point or the other was killing enough people or rendering enough people sterile that you would not that it made a difference in terms of its ability to be selected for in the genome. So, what genes are actually part of those virus interacting proteins, you might ask? And it turns out people have looked at that. And when they look at it, they show that a lot of genes that are under selection pressure in the human genome, in this particular study, they asked the question, just broadly speaking across the genome, what are the different sequences that are under positive selection pressure? So, similar thing that we talked about, they were looking for positively selected genes. And what they found was that a large number of the genes that are under positive selection pressure, which are shown in blue over here, this is sort of the overall histogram. You can see there's a chunk of genes under negative selection pressure in blue, sorry, positive selection pressure in yellow. When they dug into the positive selection pressure genes, they found a lot of them had to do with transcription, a lot of them had to do with innate immunity, transcription, gene expression, signal transduction. But there's a ton of stuff in here that is directly related to the immune system. So it goes to show you that there's a clear-cut factor that comes from evolution. We know some of this. I'm going to go through the next few slides a little faster. So human HLA genes, for example, encode a lot of diversity. Everybody has different HLA genes. And it turns out this kind of diversity is part of the Red Queen's race. We'll end up doing at some point a much deeper dive into the Red Queen's race. And in the Red Queen's race, one of the strategies is that if the host population has a very wide range of alleles, for immunity, it makes it difficult for a single virus to defeat all of them. So you see that diversity in human HLA genes that is part of that kind of evolutionary response. And it's been shown by people that HLA genes undergo coevolution with viruses. So over here, you can see the degree of coevolution, and you can see that for different genes over here, for different viruses over here, there's a wide range of different HLA loci that are under selection, both positive and negative selection. So if there's no selection, it should look white. And if it's under strong positive or negative selection, it should look either red or blue. And you can see for every one of these particular viruses in the columns, you can see that there are many different HLA alleles that are either selected positively or negatively. So that goes to show you that, again, something must have been going on in order for it to be under selection. This doesn't happen without people either dying or failing to reproduce. It turns out that heritable factors also play a big role in the effectiveness of vaccine responses. So it turns out that when you look at individuals, when they do twin studies, what they find is that depending on age, the amount of heritability and the amount of variability increases. But when you look at very young twins, what you see is that if you look at the vaccinations that happen at an early age, pretty much all of them have very strong components defined by heritability. So two twins will respond the same way to a vaccine for the childhood vaccines. For vaccines that happen later, such as Hep A B, which is 18 to 65 years old, there's much less. And when it comes to diseases like the different influenza strains, there's no heritable factor. But for the childhood vaccines, it's mostly heritable. So that's kind of interesting, right? Because what that tells you is that the way that the immune system responds to these viruses, and some of these viruses are very quote unquote mild, like mumps or measles, and still they have a strong heritable factor to them. What it tells you is that these viruses have been shaping our genome by at some distant point in our future or in our ancestral species, in our past or in our ancestral species past, killing off individuals. So yes, it's true. Human evolution will lead to lower IFRs. But what the genomic study suggests is that it does so on the basis of long time scales. And if you look at it, if you take a different lens on this, what you can see is if you list all the different viruses, like common viruses, right, and then ask the question, well, how old are they, and what is the case fatality rate? You have diseases like AIDS and sleeping sickness. I'm sorry, sleeping sickness is not a virus, but this is just in general pathogens, right? So sleeping sickness or AIDS, these are relatively new, and they have very high fatality rates. If you look at very ancient diseases such as CMV, which is not even considered a disease, cytomegalovirus, or endemic coronaviruses, these are mostly, rarely fatal. They've been around for millions of years and they don't really kill anyone. Adenoviruses, 700,000 years ago, almost 0%, but they still kill young and healthy people. But then if you notice what that cutoff is, it's about somewhere between, you know, 10 centuries and 40 centuries ago, where you start seeing these diseases having like appreciable or non-zero fatality rates. So what that suggests is that it takes a long time for a disease to really evolve to drive human evolution to the point where it's not necessarily that lethal. So we've talked about pathogen virulence, and there's very little evidence that shows that that happens. But give it enough time and mortality, 40 centuries from now, I'm sure we'll all be resistant to COVID, or our descendants will all be resistant to COVID if we pursue this strategy. You just need a little bit of time, if you know what I mean. Uh, so coming back to this, like, you know, what it comes down to is that red queen's race happens at two different levels. You have the acquired immunity level and you have the evolved resistance level, as we talked about, right? And so acquired immunity happens on a relatively fast time scale, evolved resistance happens on a much slower time scale, right? And so when we think about it this way, um, when viruses or when pathogens gain virulence during emergence, we've talked about it, then you end up with a fitness advantage. People have looked at this with polio, for example, and what they found is when oral polio vaccine viruses, oral polio is an attenuated virus, which people were given in the 80s. When oral polio vaccine viruses occasionally revert, and they study the DNA of those oral polio viruses, what they find is that those viruses will often show an increased virulence. Um, so gaining virulence can be advantageous. And this particular study shows that. And what it showed was that essentially when you have these reversion mutations, you end up, if you take those reversion mutations and you put them into susceptible mice, you end up with essentially killing the mice much more efficiently. So this is sort of a drawing that shows that essentially what they showed was that the oral poliovirus, in order to get to the pathogenic version, ended up climbing a fitness landscape, so to speak, and ended up becoming more virulent. So it happens in real time as well. Another factor to consider is that global connectedness also favors virulence evolution. So we've never been more connected than we are today through shipping, through airline networks, and so on. And people have done studies, for example, this computer simulation study, where they ask the question, if you're more connected, will you be more virulent? And so over here, you see these little diagrams of the simulations as they're running over time, as it's evolving, 20 generations, 6 different runs, 8 different runs. And what they did was they asked the question, what is the threshold level at which a virus is sufficiently virulent that it collapses the population? And they found that that threshold level where it switches from being um, able to coexist to not being able to coexist at all, that threshold level varies. And so here they looked at two different kinds of networks. They looked at small world networks and they looked at networks where there were not a lot of connected hubs. And they found that in the top panel in A, what they showed it was for different viral growth rates and different levels of virulence, small world networks led to population collapse much more easily, which is shown by these numbers, than in sort of networks where that was not the case. So essentially, once you have small world networks where you have hubs, such as airline hub, those hubs actually facilitate rapid rapid transmission of disease that can even kill locally. So complete mixing results in rapid global extinction, which is what you saw in the small world networks. while the spatial systems achieve long-term stability because local extinctions don't immediately spread. That was their take home here. Keep in mind, as we've talked about, that humans are still running the Red Queen's race. So if you look at malaria, for example, malaria has been a very strong force of selection in humans in the past past tens of thousands of years. So mosquitoes emerged as a human parasite about 10,000 years ago. And if you look at where malaria is endemic and where the sickle cell anemia allele um, occurs, there's a very tight correlation. Right. And so as recently as 2005, there was 1 million dead a year from malaria. And often it's a lot of children. So in the era before quinine, about 50% of children in Africa and Asia would die of malaria before the age of 4, which is really staggering. Now, different populations have evolved different evolutionary responses to malaria, but the fact that the sickle cell allele confers resistance is kind of interesting. And I'll show you why. If you look at this, across a different range of genes, there's different kinds of genes that protect against malaria. So if you look at, there's one called the Duffy antigen, which is a chemokine receptor. There's a sickle cell anemia gene. And so you can actually look at these genes and ask the question, how quickly does evolution happen? So they actually did this in Cape Verde Island using the Duffy antigen receptor, which is one mechanism for resistance. And it turns out that this is almost fixed. Almost 100% of people with West African ancestry have it, and other people don't have it. And what they found was, if you look at over time in the Cape Verde Islands, these 3 islands over here, on the 3 different islands, what they found was that the enrichment of West African ancestry at that particular locus, so in other words, genetic introgression, as we saw with the Neanderthal human case. So because these are Cape Verdeans are sort of a mix of people with local ancestry and West African ancestry, the picking up of genes from West African ancestry indicates selection or introgression. And what they found was this got picked up very quickly over a set of generations, at some point. And what they found was that that happened in over about 500 years. So they found was in this particular population that had a mix of people with West African ancestry who were completely resistant to, or who had a strong level of resistance to malaria versus people who didn't. What they found was the introgression of that Duffy antigen happened over a period of 500 years. So again, malaria kills about 40–50% of people who are not resistant. And so over a period of 500 years, when it was that high a level of death, it still took 10 plus generations, 20 generations for this to work out. So if you look at generation 1 and you look at the amount of the antigen versus generation 20, you can see that there was a clear shift, but it took 20 generations. So again, it takes a long time. It kills a lot of people, is the bottom line. If you look at the map of Africa, interestingly enough, from 1880 to today, what you can see is that European colonies in 1880 were limited to the coasts. And by the time, quinine came around, right around that time, it changed the game, because prior to that, any expeditions up the Congo essentially ended with everybody dying, or nearly everyone dying. They failed repeatedly. But with quinine coming in, that was not the case. Now, Liberia was founded successfully in the same era, and Liberia was a population of returned folks who were descendants of enslaved people or enslaved people themselves, and so Liberians managed to set up a colony in West Africa, and be able to hold on to that colony and expand it, as the case may be. But that was not the case for Europeans who were exploring Africa at the same time. So with quinine, by 1913, the game was completely different, and all of Africa was colonized. And so that's kind of interesting, because what it goes to show you is that the genetic factors and evolution of disease resistance can have a huge impact on the way things play out. The Red Queen's race with malaria is still ongoing, because malaria itself is evolving in the other side. It's evolving resistance. And so taken together, what this means is the humans run the Red Queen's race all the time. We are shaped by it. The story about colonialism shows you that it even impacts our history, it impacts our genetics, it impacts our ancestry. And evolution is a major shaping force, but it's very, very impractical to then manage diseases using human evolution because it involves huge numbers of people dying over extended periods of time. It's a very silly way to manage disease. Public health's job, in fact, should be to prevent evolution. And when you try to rely on antagonistic coevolution to manage an emergent pathogen, that is just not clever at all. Um, and the bottom line is that this kind of an approach can lead to very unpredictable outcomes. And this is why we don't try to domesticate viruses. Even diseases like adenoviruses, which are supposedly benign, quote unquote, are benign because we paid a price in blood for that. And they can change, the virus can change the terms of the deal on us at any time. So at the end of the day, this is not a practical strategy. So summing this up, novel pathogens can lead to deadly pandemics if the virulence starts out or evolves to be high. Increased virulence is beneficial or neutral to the pathogen, as we saw. There's no experimental support for stable attenuation. It is true that hosts evolve resistance, and therefore most diseases tend to become milder over time, but that happens over many generations, dozens of generations to hundreds of generations, depending on how strong the selection pressure is. When you have something as deadly as malaria that killed 50% of infants in the Cape Verde Islands, it happened over 20 generations. Um, so it's really a host phenomenon, and evolution is not our friend, as we discussed. It's a relationship of antagonistic coevolution, not mutualism. And running a Red Queen's race with a newly emergent pathogen is a very silly idea. It's an objectively silly idea. So that wraps up this particular presentation. In the next talk, we'll look at what history teaches us about pandemics, and we'll look at sort of how the historical context with more recent pandemics. A lot of these things are in our genetics, in our DNA, and in graveyards. We'll look at more recent examples and ask the question, what does history teach us, and how can we infer things from that?
Very interesting. I know we're going to be moving into some of the rest of the topics, so I maybe won't ask too many questions or make too many comments. But I just want to note that one of the things that I often get asked from students taking a course in evolution is, are humans still evolving at all? And the premise behind that, I think, is, well, we have medical interventions now, better diets, and all that sort of thing. And the response is, some of us have access to all of those interventions, and billions of people don't have
Yeah.
access to those. So there's still huge numbers of humans that are not benefiting from those protections, treatments, those sorts of things. So it's a little bit of a positional fallacy, I guess, to sort of think that all humans are experiencing the same conditions that Western, well off countries are. The other is, there's some interesting. There's been an interesting argument about rates of evolution in human lineages over time, and one of the things that comes up again and again as a theme with these sorts of things is uh, whether the selection pressures are changing, but also population size is a huge, huge factor
Hmm.
for a number of reasons.
Yeah.
So one of them is more individuals contributing to the gene pool means more mutations being added to the gene pool, so you've got more variation to work with. The other is natural selection is stronger in large populations than in small populations, and so the larger the population, the more likely selection will be observed over something random like genetic drift. Well, human population size has exploded, absolutely exploded in the past couple of centuries, really. But also the shift to agriculture has not only enabled some of that increase in population size, but has dramatically shifted what selection pressure. So dietary, but also exposure to zoonotic pathogens through domesticated animals and that sort of stuff. So the
Yeah. Yeah.
argument has been made that, in fact, human evolution over the last 10,000 years or so has been faster than any period
Yes.
in our
Yes.
history because of the things like that. So I would say, even, I know you're going to get into history, but for folks who have been wondering about, okay, what are the prospects for human evolution now, and do our protections remove us from that sort of red queen in a way? I would say no, because we don't all have access to them. And secondly, that even compared to something like 1918, the flu pandemic, there are many things that have changed, not in our favor, as far as these kinds of processes. So population size is massively larger. It's
Right.
like 4 times as large as it was. Global travel, all these kinds of things. And so we, in terms of, is evolution still an option? Is evolution still a potential outcome of these arms races for humans? Absolutely, and that's not a good thing.
Yeah. So the article that you and I and Lynn Theatre wrote for
Mhm.
Scientific American, we made the point that if you compare it point for point, we are worse off today than we were in 1918 when it comes to influenza.
Mhm.
Um, Literally, modern medicine has not made that much progress against the three major ways in which influenza kills. So if we had in which pandemic influenza kills, so if we had pandemic influenza tomorrow, you can expect precisely the same kind of negative outcomes that you saw 100 years ago. It's not moved that much. And in addition, as you said, it's a much more interconnected population. Diseases will spread much more quickly. In theory, we have public health, but public health has gone quiet now. Public health has given up. So we don't actually, even with this current influenza pandemic, we don't have contact tracing going on. So we are very much at the mercy of infectious disease as a conscious choice and decision made by governments worldwide. Every government worldwide has made this choice. And the point is, if we are planning to try to live with infectious disease, we are planning to allow evolution to take its course. That will happen over centuries, even if disease is extremely deadly, as was the case with malaria
Mhm.
in the Cape Verde Islands. There is no rational way to use evolution to manage the disease burden in a population. There's no rational way unless you're shooting for human extinction, which, by the way, is a possibility. The only way to make infectious disease not a problem in humans through evolution is to let humans go extinct through it. Otherwise, it's going to become more and more of a problem. It's going to be more of a burden on our systems, not less.
Mhm.
And the other thing is when you talked about the human evolution, the if you think of sort of gazelle evolution, right? What drives gazelle evolution? Largely, it's the Red Queen's race with cheetahs, right? So cheetahs and gazelles are driving each other's Red Queen's race evolution because one eats the other and vice versa, right? So what is the thing that eats humans the most? It is infectious disease. So one of the biggest biotic factors for human evolution is disease. In fact, it is the biggest biotic factor for human evolution. Our apex predator is infectious disease. So in fact, infectious disease is well documented that infectious disease has been a huge issue ever since the transition to agriculture. So when humans settled down about 100,000 years ago, about 10,000 years ago, that was when infectious diseases exploded because they were in sedentary populations, there was large populations, all the things you talked about.
Mhm.
So, in fact, disease is our primary predator, and infectious disease, except for the 20th century, was exerting huge amounts of selection pressure on humans, as is well documented in the literature. That statistic I told you about half the children dying before the age of 5 in Africa in the 19th century, that was true across the board through pretty much all of human civilization. Romans had similar numbers. The Chinese had similar numbers in Ming China in the 19th century. So this has been the case all the way through. And so if we return to a point where we're not controlling infectious disease, A, we will need to accept the fact that half the children in our societies die before the age of 5. And B, we can't necessarily expect any immediate benefit from that huge mortality toll.
I think one other really important point, and then we'll probably wrap this episode up, is the other argument that you often hear is, well, humans have coexisted and co-evolved for millions of years alongside, respiratory viruses like this and this
And
and that.
that's true.
It's true to an extent, but not in the way that we are now. So when
Right,
people say that, they're thinking of bats, right? And
right.
the way we encounter them now is not how we have before. We were, previous up until agriculture, in relatively small groups, primarily outdoors, moving around, not living next door to,
Yeah.
domesticated livestock, all those kinds of things. And those have all massively changed since agriculture, and in particular in the last couple hundred years, where we spend huge amounts of time indoors at high density. Bats have lived like that in caves in huge numbers, and they have particular adaptations to those encounters with respiratory viruses that we do not have. And so it's also a
Right.
myth that we're already kind of set up to deal with this. This is a
Yeah,
novel,
no,
evolutionarily a
100%. We're
novel.
absolutely not, we're not set up to deal with any infectious disease. That's not how it works. Um, that's not how evolution works.
Yeah.
I think what happens is there's a lot of sort of what one of our collaborators, Randy Ness, calls tacit creationism. There's a lot of sort of God will provide reasoning. It's like, oh, let evolution deal with it. Well, A, evolution is not your friend. It is messy and it is random. And B, evolution involves killing or rendering sterile large chunks of the population. So it is not the same thing as the benign hand of God intervening on your behalf. And I think that that creationist mindset kind of is in the back of a lot of people's minds when they say things like that. It's like, no, you have to understand that it is absolutely public health's job to slow down and stop evolution. Very much in our case, evolution is the enemy because the societal an economic price that we will pay for evolution is not one that we could bear easily. Bats, when they are subject to evolution, white nose fungus, it wipes out entire populations, colonies of bats like that.
Mm-hmm.
Certainly, if we had a situation where the entire, city of Las Vegas went under because of some rapidly spreading sexually transmitted disease, that would be most of us would consider that to be a very bad thing. So
Yep.
at the end of the day, I think we're not configured to lose whole sections of the population because of, the biblical hand of God and some plague coming down, on the first one. That's not how modern societies work. And so accepting this as being, well, this is fine,
Mm-hmm.
is just incredibly silly. It's not compatible with Western civilization to take an approach like this, or any civilization. It's not compatible with any large societal structure, and it's not compatible with an interest in the continued survival of a species or any species.
Mm-hmm.
If you say, well, let infectious disease go unmanaged.
Absolutely. So I'm looking forward to the next installment where we're going to hear more about historical examples, I assume in humans. But this is a very good biological context for how all of that plays out in our own species as well. So thanks, Arijit, for a very interesting talk, and reminder to everyone else to check out some of the other videos on the channel. There's a whole series on cancer that's well worth a watch, and we'll provide some links in the description to Scientific American article and a few other things that were mentioned here, the commongering piece. And we'll see you next time.
Great, thanks, Fred.